Yes, okay. So today I'm going to present the work I did when I was at Harvard University working under the supervision of Radhika Nagpal and Justin Werfel. And which is, studying how the surface guides the construction in mound-building termites. And then when talking about the mound-building termites, I'm mainly going to talk here about Macroternis mycoseni. I'm going to make one small comment about Macroternis natalensis, but then Macroternis mycoseni are these termites found in Africa. We usually go to Namibia to study them, and they can create these mounds. We can reach 3-4 meters of height. Here is an example of a typical one with my boss for reference of the size. And the outer structure of the nest is actually like a big chimney, as we've seen in previous talks. So this is responsible for the ventilation, gas exchange, and all that. And all the interesting or essential structures for the nest is actually underground, right? With the royal garden, with the galleries and the nest and broods and everything else. Our main focus when we were studying termites was actually to study stigmergy, right? So stigmergy is this environmental effect that will attract activity of some sort, right? And then very well-known hypothesis in especially termite construction, but also in ants, is the cement pheromone, right? So then it has been long proposed that by depositing certain structures, the termites or ants will actually add a pheromone to this, and this will attract more and more depositions in the same locations. And then this would cause these pillars and ceilings to arise. So far, we have not been able to find any evidence of cement pheromone, but we have been able to find evidence of other stigmergic properties. All right, so I'm going to focus in two. I'm going to just give a brief introduction of how humidity affects termite activity, and I'm going to highlight some of the experiments I did in these two papers here. So it's just like a very fast summary of how humidity also acts as a stigmergic factor into mound construction, right? So basically our hypothesis was that termite mounds repair and construction expansion is related to this humidity threshold, right? So when you have a sunny day, what we measured that actually the humidity was dropping very fast as it left the mound. So then if you make a hole into the mound during a day or when it's very windy, then the termites will only repair and close this structure. But then if this happens at night, you can measure this profile of the humidity and you see that it actually extends a little bit outside the nest, and this would lead to the expansion of the nest. But then to better study how humidity would play a role into the construction of termites, we developed this humidity chamber, right? So it is a box with a humidifier in the bottom, right? And then some humidity sensors. So the humidity doesn't, there's no direct contact between the jets of humid air with the termites. And then you have a cartridge of solid mud with a disc and a hole in the center, right? And then we are recording all this from the top with an Intel RealSense, which give us RGB images, but also this depth imaging of the mound, so we can see in real time how the mound is constructed, right? So when we place the termites on top of this disc, they can only hide by digging through this mud cartridge. And then we are separating the two stimuli of digging and constructing. So everything we see on top is their construction behavior, and then we lose all the digging. But in this way, we can focus in actually how the termites are building these structures without being connected to their digging behavior. And then it's not a surprise that when you have high humidity experiments, they build more, so they create larger in area structures. These structures are heavier and they also have a higher volume. But one thing that was very interesting for us is that if you compare these two termite species, and then in Namibia, they live side by side, you have Microterminus microsanii with the very tall mounds, and Microterminus natalensis with these short but a little bit stubby mounds, right? And then so far, everybody has tried to actually identify what kind of behavior would drive the termites to build differently, right? But they seem until now to be indistinguishable from each other. And then what we saw is that when you do experiments that you would think like, okay, we're going to do experiments where the termite is comfortable, high humidity conditions, the closest to what they have in the mound, there is no difference between the two, right? As everybody else found. It's only when you control the humidity environment and you place them in a low humidity environment that you can actually see consistent differences between these two species. Right, so here it's an example of how looking by different humidity patterns that we were able to actually separate these two species into different behavioral aspects. But now going to the main topic, how curvature affect the construction. So the initial idea was actually to see how topographical properties would affect the construction of termites, right? So we know that termites, everybody who has worked with termites know they really like edges, pillars. They'll go to these regions and dig and build around them and create arches. So what we did was we created this borderless Pringle-shaped structure, right? And then it's formed with a concave part here on the top, if you can see my mouse, and then a flat region over there, and then a convex part here. All this structure is perforated so that when we cover in mud, the humidity doesn't pool into the concave regions, right? And then we can place termites on top of this and make experiments with different inclinations, right? The different orientations of this structure. And then by knowing the curvature, which is invariant to rotations, and the inclination of the treatments that we propose, and also the height of these shapes, because the termite might be integrating, oh, am I on the top of the structure, am I on the bottom of the structure? If any of these variables would affect their behavior, right? So these are some videos of the typical activity of the termite in these experiments. So here is like 3 different videos from 3 different colonies in the same treatment. This is what I call horizontal treatment. And then we can see that termites very fast, this is a 1-hour experiment. They go to this same region, which happens to be the concave region, and then they start digging and depositing around that region. By using some computer vision techniques, we can see what were the areas where most activity happened. And then here we are seeing activity in general. We cannot disentangle digging activity from deposition activity, right? But in our experience, deposition always happens at the border of digging. But the resolution of our videos didn't allow us to actually disentangle these two properties. And then, as I mentioned, we designed this structure, so we have all the information possible. So then now we can see how the termite soil displacement activity compares with the height, with the inclination, and the curvature in these 3 different rotations, right? And we also track the termites for the first 5 minutes of the video. Why 5 minutes? Because this is before digging started. And then we know like where termites were walking without being influenced by the digging activity of previous termites. And then here you can already see that there is like a very good agreement between termite activity and location with curvature. But to be a little bit more quantitative, we did the correlation of all the different treatments and all the different topographical properties. And only in curvature, we had a consistent correlation between termite initial position and the soil displacement, showing that yes, termites do prefer these concave regions to start their digging process. I would like to thank my colleagues, so Nicole Carey, who is responsible for building every one of these devices that I used in the field, Paul Bardunias and Rupert Soar, who were in the field with me, helping with the experiments and giving overall assistance, and Scott Turner, Radhika Nagpal, and Justine Werfel for the supervision. Thank you.
Many thanks.